you, Roy, and thank you, Amit, for hosting me in this venue. I'm certainly not the standard speaker in this kind of seminars in your faculty, so I want to thank the faculty also for the opportunity to speak here. I will be talking about the work I've been doing for the last 4 or 5 years in my PhD, mainly involve synthetic DNA libraries and what we can do with them. And as I already said, I've been around the Technion for a while now, almost 12 years now. So let's start a little bit with the introduction of myself. I started in industrial engineering, and right to around the end of my first degree, I decided to start a research project in Itai Nai's lab in the Faculty of Biology. This research project actually turned out to be my master's work at the end, where I did some bioinformatics. We mainly worked on single-cell RNA-seq and examined different systems of evolution and development in different animals. I then took a year off the Technion, although not quite, since I kept teaching at the Technion this time, but I was working for Microsoft for about a year, where I did some machine learning work about recommendation systems to content for Microsoft customers. This is where I actually met Professor Zohar Yachini, who was my PhD advisor. And we started working, and we immediately started a collaboration with Roy, which continues up to these days. And I will be presenting the work from this work in this talk. And to understand maybe how we came into this special world of synthetic DNA, I come from a very diverse background. I have a strong background in computer science, in information theory and coding theory. I also have a deep understanding of statistics and machine learning. And to complement it, I have, I think, some experience with molecular and cell biology, although I never held a pipette in my hand before. In the intersection of all those 3 fields, we can find the field of synthetic DNA libraries, where we do various projects integrating, let's say, concepts from all 3 fields and tools from all 3 fields together. So what we're going to be talking about today, I guess most of you don't really know about synthetic DNA libraries, so we'll start by introducing the concept of DNA synthesis and synthetic DNA libraries. We'll then move to describing one project in which we used synthetic DNA to store information, digital information on it. And then maybe switch gears to a more biologically oriented project in which we're gonna talk about how we use synthetic DNA library to study the mechanisms of specific gene regulation mechanism in E. coli. Okay, so what is synthetic DNA? Those days, I think we all know this picture from the coronavirus swab test that you all get. What happens to the swab later on is that it's going to be sent into a PCR test, as they like to say. The PCR test is used in order to identify the genetic sequence of the virus in the sample. For this, they use synthetic DNA as a primer for the PCR. I'm giving this introduction, although I know most of you from the biotechnology faculty know it, since my mom is also here, and I want her to understand as well. So, this is a bit to get her up to speed with everyone. We're going to use synthetic DNA is used in order to generate DNA molecules with the desired sequence that we want in order to use it later on in biological assays or in diagnostics or in any other biological context. And how do we get those synthetic DNA sequences or molecules? So most of you students probably know this page of the ordering oligos from IDT. You design your sequence. That's what you see here on the left. You design your sequence. Then you send the sequences that you designed into a vendor. In this case, this is IDT, but there are various vendors that can generate this service. And then a few days later, maybe if it's a complicated request, a few weeks later, you'll get to the lab a tube containing the molecules of the DNA molecules with the sequence that you designed. And this is very nice if you want to do something that involves 5 or maybe 10 or maybe even 20 different sequences. But if you want to scale this up and get a very broad range of sequences, this will not scale up. This would become very expensive. And in this case, what you get is the concept of oligo libraries. This is what we see in this slide. This slide represents the concept of ordering a large amount of different DNA sequences. The procedure is very similar. You then send those sequences into the vendor, in this case, Twist Bioscience. Then you'll get the pool of DNA molecules in the lab, same way you did before. The one difference is that in this case, instead of a few dozens of sequences, you can order a few hundred thousands of different sequences, and they're all going to be manufactured together, and you're going to get them all together. And in order to understand how it's done, let's see a little example of how DNA synthesis works, or how large-scale DNA synthesis of DNA libraries work. So what we see here is a very general description. Usually I present here a movie, but because we switched into a Zoom venue, so I'll change it into this nice comics. So what you see in this comics is the different steps of synthesizing DNA. On this slide, which is a very small slide, you get many different spots. In each spot, you're going to synthesize a different sequence, a different DNA sequence, and it's going to happen in a very similar manner to how inkjet printers work. So in this case, it's going to be an iterative process in which in each iteration, the next DNA base or the next DNA letter is going to be synthesized onto the elongating molecule. In this case, you see that in the first step, we want to synthesize A in the beginning of the sequence. So we basically inject A's onto the surface and they elongate the molecules. We go on, and then at the end, we get the desired sequence in multiple copies, in which we can then harvest from the slide and ship to the customer, which is us. A few interesting or important properties of this process would be, first, that since this is an iterative process, the number of iterations that you can do is limited. It's limited, and currently the current limitation of the technology is somewhere around 200 to 300 cycles. And we have to keep it in mind because some of those properties are going to come back to us later on as properties that we have to deal with. Next, the number of different sequences that we can synthesize in parallel on the same slide is somewhere in the range between 10,000 sequences to 1 million different sequences. And as we saw, we're going to get many copies of the same sequence synthesized in parallel in each one of those spots. So we're going to get thousands of different sequences, each one of them in thousands of copies, all mixed together in one pool, in one tube. And we have to remember that once they're mixed in this tube, we cannot separate them unless we have a way unless we took care of it in the design, let's say. Okay, so now I guess that we're all up to speed with what DNA libraries are. there are very different and a very wide range of applications that we can use DNA oligo libraries for. I will describe two. We'll start with the DNA data storage, and then at the end we'll get to something which is more biological. So,
how uniform are these libraries? How uniform?
That's a very good question. So the uniformity varies between the different vendors, and of course you cannot expect it to be very uniform. But let's take Twist as an example. Twist is the leading vendor today in the industry, and they generate quite uniform libraries. So you do not see too many outliers of sequences that overcome the entire library, and you can expect to see all of your design sequences present in the output. Also, of course, there are other limitations that sometimes are sequence specific. Some sequences are harder or easier to synthesize. Most vendors have this quality control step before the synthesis, where they see your design and they try to assess how well can they synthesize this, and maybe try to tweak your design a little bit so it will be easier to synthesize. But that's also important to keep in mind that whenever you use oligo libraries, you should keep in mind that some uniformity is lost throughout the process, or there is noise in the process, and you have to maybe control for it in the different scenarios that you use it. And we try to do it. So we developed tools in which we can assess the uniformity or the quality of the library that we have in hand, and maybe use it as a checkpoint for moving on to the next step of the experiment, for example. Okay. So data storage. I guess these days also using the term exponential growth is also very common and very popular. So what we see here is the exponential growth of digital data in our world. This is an estimation from a report that was published a few years ago about how many data are we using or are we generating and storing in the world. And the exponential growth is the concern, since we all know what happens to exponential growth if we don't flatten the curve, and we do not want to flatten the curve here. We just want to be able to store all this information that we're generating. But this is a very important, maybe let's call it motivation to develop new ways to store information. And one thing that we all we know is that, just to give you like maybe a hint about how much information is this. So what we have today, or what we was expected to have today, is somewhere around 40 billion terabytes. So 40 billion hard drives of 1 terabyte. This is a lot of information to store, and it's raising concern about how to do it. The flip coin of this problem is what you do with minimizing the storage, like the volume you need in order to store all this, and also the energy consumption of this. So what we see in this slide is a very nice classic pictures from IBM from the 50s. And if you wanted to guess how much information can you store in this large box, it's actually a 5 megabyte hard drive from the 50s, from just 70 years ago. This is what you needed in order to store 5 megabytes 70 years ago. The downside of this is that we're nearly at the end of our ability to actually scale up or minimize anymore the storage media. So we do need to look for alternatives. And DNA is maybe the natural alternative. So what we see is that if we look at DNA in nature and we ask how robust and efficient it is in storing genetic information, we can see that the most efficient organism that we know today does it 12 orders of magnitude more efficiently than the current state-of-the-art magnetic tapes that we use. And if we consider the robustness of DNA as a storage media, so whenever you have a magnetic tape that you use in order to store your information, you have to take care of it every 5 or 10 years. You have to read it and rewrite it again on a new tape in order for it to be still viable, for information to be still viable. While DNA, we know that we can store for many years, thousands of years, and come back to it after thousands of years, buried in the snow, and read the information of it with very little errors. Okay, another maybe important or appealing property of DNA is that it's not going to become obsolete anytime in the near future. Or as long as we're here, it's not going to be obsolete. What you can see here in this slide is the very different media or media types that were used in order to store information in the last, let's say, century. We all know the old VHS tapes we have at home from our, let's say, bar mitzvah or birthdays as kids that are practically useless today because we have no VCR machines to read them. DNA, on the other hand, is the same DNA that we used to store genetic information billions of years ago, and the machinery used to read and write it in the cells is the same machinery. The same enzymes or very similar enzymes that are used today were used 3 billion years ago. Okay, so DNA is a very attractive alternative as a storage media. How do you do it? The process of storing information on DNA is described in the slide. What you see is that you start off with a binary file, and in this point, maybe I'll give just the comment that we know that everything we have on our computer is stored somewhere as a binary file, as a sequence of zeros and ones, as we can see here. Then you need to somehow encode or translate this sequence of zeros and one into a sequence of A, C, T's, and G's, which are the building blocks of DNA. Then writing those sequences, we already took care of because we can write it as an oligo library synthesis. We can then hand it over to Roy's lab and just tell him, okay, Roy, maybe you can store this in the fridge for me for a while. I will come back for this information later on when I want. And then a few months later, or maybe after I come back from my postdoc, I can come back to Roy's lab and ask for my tube back and send it into NGS DNA sequencing, which will help me to read the sequences of DNA stored in the molecules in this tube. I can then use the same mechanism, or maybe the complementary mechanism for the encode in order to decode this information and get my binary file back. This completes the process of taking digital information, encoding into DNA, storing it as DNA molecules, and then reading it and decoding it back into the digital information that I want. Just to give us a very quick overview of this field, this idea is not very new. In the last decade, there have been, as you can see here in this slide, some quite of action in this field, and you can see that in each one of those examples, I gave the total amount of information that was stored and the logical density that we're going to define in just a minute. That was demonstrated in this work, and you can see that as we go along, we manage to store more and more information. So we can scale this up a little, and we can also get to a much denser or much higher logical density, which means we're also improving in terms of efficiency of the system. And now, just also to give us just an understanding of how much information can we actually store in this in DNA, if we take the equivalent of one grain of sugar in DNA, this will contain 1 grain of sugar. This will contain 125 terabytes of information. A few hundreds computers stored in 1 grain of sugar. If you take 1 teaspoon of sugar worth of DNA, it can contain up to 800 petabytes, which is basically all you have in YouTube. And if you just fill up a whole truck full of DNA, you'll get all of the world's data stored in this one truck. So, and those numbers are the actual numbers that we see. So this is the actual density that we can achieve. It's not just a theoretical one. We cannot scale it so much at the moment, but this is the actual density that we can achieve. Okay, if we move on to how we encode the information in DNA. So we said that the first step would be to take the binary file and maybe translate it into a sequence of bases, A, C, G's, and T's. The maybe most naive way to do so would be to say, okay, we have a binary alphabet of 2 letters, 0 and 1. We have a DNA alphabet of 4 letters, A, C, G, and T. So let's just take every pair of bits and assign a specific letter, a specific DNA letter to them. So 0, 0, 0 will become A, 0, 1 will become C, 1, 0 will become G, and 1, 1 will become T. Now, if I'm given a sequence or a binary message or a binary sequence, I can then split it into pairs of bits. Each pair would be translated into the correct letter using this table, and then I will get the output DNA sequence, which would be half as long as the binary message. This can then be taken into sent into synthesis. I will get those molecules of representing this sequence synthesized for me, which I can then store, sequence them, get the sequence again, and decode. And now we want to ask, what happens if we have more than 8 letters? More than 4 letters. What would happen if we had 8 DNA letters? And this, just keep that in mind for a while. We'll come back to this in maybe 2 or 3 slides. Okay. Do you remember that we discussed about, we talked about the limitations of DNA synthesis or the oligo-library synthesis? And the first limitation we mentioned was the length, the molecule length. We're limited to somewhere around 200 or 300 bases. So what this means, means that we have to break down our binary file into separate lines, and each line would be then translated into the correct DNA sequence, which would be short enough in order to hold all of it together. And then another thing that we said, that all of those different DNA sequences are going to be synthesized together. In order for us to synthesize all of them together, what would happen at the end is that we're going to get them all in one tube mixed. In order to recover the original file, we have to remember what was the order of the different sequences. And for this, what we do is we add those indices. Indices, or what we have to like to call them, the page numbers of the messages. So each one of the binary lines would have an ID, which would also be translated into DNA and synthesized together with the information. Then whenever we sequence the information, we sequence the address of it as well, and we can rebuild the file at the end. How do we measure the density or the data density of DNA-based storage? So we'll discuss 2 different density measures. The first one would be physical density, which is what we described so far. How many bits can I fit in each gram of DNA, or how many files can I put in my tube, for example. And as we saw, this in today's technology, using today's solutions, we can get orders of magnitude larger than the magnetic tapes. And the other maybe density measure would be logical density. The logical density would be defined as how many bits can we write in each step of the synthesis, in each cycle of the synthesis that we saw. The one thing to remember is that we pay for synthesis for each cycle of synthesis. So this would be the correct way to measure the overall cost of writing the information. And one thing to remember also is that we have 4 DNA letters. So in each cycle, we can decide on which of the 4 letters to write. And we'll get 2 bits, which is the 1 letter that we write in this cycle, per each cycle. This is the theoretical limit of logical density as it was known up until our work. Okay, this brings us to describing our work. So we covered all the background of storing information on DNA. So whoever is only interested in the general idea of storing information in DNA heard what he needed and can now leave if he wants. Now let's describe our work. So this work was published just recently and is the output of the very fruitful collaboration with the lab of OE. And let's dig into it. Okay, so
before you start, what is the error rate? Because I think this is probably very important, both in reading and writing.
Yeah, so let's start by defining an error. So we can maybe talk about 2 ways to ask if we have an error. We can start by asking, can we fully recover the file that we wrote in the DNA? And this requires us to either be able to fully recover each one of the parts that we wrote, or to include some kind of an error correction code over the original information, which will allow us to overcome few or as many errors as we can design. But where do we get errors from? So we get errors from synthesis errors. So sometimes we will ask the company to synthesize C in a specific position, and some of the molecules will just get an A. And then this is what we're going to get in the tube. And errors can come also from sequencing errors. So the molecule actually contains the correct letter C, but when we read it, we get the incorrect error, the incorrect reading of, let's say, an A. The real way to deal with these kind of errors is to keep in mind that errors usually don't come on all of the sequences together. So if we read enough copies or enough molecules that represent the same sequence, the consensus of them would be the correct letter that we wanted to write there. And usually this is and since we synthesize many copies, which are independent and do not share errors between them, hopefully, so we can overcome those errors. But if we do want to talk about the error rates that we usually see, so in synthesis, we get mostly deletions of bases, and this happens once in every 2,000 bases, let's say. And in sequencing technologies, we get mostly substitution errors, which we can see also 1 in 1,000, 1 in 10,000. It depends on the quality of the sequences we get. But usually in DNA storage, we just read enough copies in order to overcome the errors. So I hope this answers your question. Okay, so what if I could ask Twist, the manufacturer, to take this 4th position and not synthesize A in this position and not synthesize C in this position, but rather synthesize some kind of a mixture of A's and C's. So this requires them to have this one specified printhead, which contains pre-mixed solution of A's and C's nucleotides in this printhead. And whenever they get to the 4th position, then you will just inject the solution from this specified printhead, and we'll get some of the molecules will elongate with an A, some of the molecules will elongate with a C, and when we get to the final output of the synthesis, some of the DNA molecules that we'll have will contain A in the 4th position, and some of them will contain C in the 4th position. Now imagine that I take this very complex wording of some of them will contain A, some of them will contain C, and I'll just replace it with just one term, a composite letter. Let's call it M. That the meaning is a one-to-one ratio between A's and C's in this position. So the question is, can we actually read this specified composite letter M? So we have a mixture of molecules. Some of them contain A, some of them contain C. When we sample them and sequence them, the output that we get, some of them will contain A's, some of them will contain C. In this case, let's say we sequenced 4 copies. 8 copies, 4 of them contained A, 4 of them contained C. I saw this special ratio of A to C's. Then I said, okay, this position was intended to be an M. So now I have a 5th letter in my alphabet, M, which is not A, not C, not G, and not T. It's a new letter in my alphabet. So remember when I asked you what happens if I have 8 letters? So now I actually have 8 letters. I introduced 4 more letters, which are composite letters of one-to-one ratio of the different bases. And now I have 8 letters in my alphabet. So now I can build this extended encoding table in which every 3 bits are going to be translated into one composite letter or standard letter in this case. And if earlier we had 32 bits that were translated into 16 letters or 16 synthesis cycles, now we can have 33 bits translated into 11 letters, which are 11 synthesis cycles. So we reduce the number of synthesis cycles we need in order to store the same amount of information in this case. One difference between the standard DNA and the composite DNA is that in standard DNA we just need to identify the base, and in here we need to identify the ratio between the different bases in the position that we read. Okay, but identify or inferring this one-to-one ratio was easy when we only had the 3 options of C, A, or maybe M, which is a one-to-one ratio between C and A. And we also got a precise one-to-one ratio in our readout, which was 4:4. But what happens if we, let's say, read 100 copies? We get 55 C's and 45 A's. Now we still have to choose which of the letters this came from. In this case, it's easy to say that it came from an M, a one-to-one ratio, with some noise in it. But what would have happened if we had many more letters to choose from? So in this case, at the end, we can see that there is C, there is A, but in between, there are 9 different letters that contain the different ratios between A's and C's. And if we get 55 to 45, in this case, it's very hard to decide if it was supposed to be 50-50 or it's supposed to be 60-40 in this case. So the inference of the correct letter becomes more complex whenever we move into higher resolutions. And this will come back at the end when we'll see the results. Okay, so what is the benefits from this solution? We increase the logical density, as we said. We drastically reduce the overall cost, but we pay with the reduction of physical density. Because now, in order to actually identify the ratio of A's and C's, we have to sequence, or we have to have more copies of more molecules representing the same sequence in our tube in order to sequence them all and get the actual ratio. So we can't just have one copy of this sequence. We'll not be able to determine if it's an A, a C, or a combination of A's and C's in this case. So there is a trade-off between physical density and logical density in this case, which we're going to see in a minute. Okay, so
what about the distance between this kind of degenerated letters? Because you have to identify those, and I guess there is some limitation on how close you can put them.
Yeah, so what you're referring to is exactly the limitation that comes from maybe moving into higher resolutions. And the last slide of the results will see maybe some sort of, we were trying to assess the ability to move to high resolutions, and it's definitely a challenge, maybe even a challenge that will make us shift into a different paradigm at the end. I will not have time to discuss the solutions for this, but you're actually right when you pinpoint the problem that it's hard to synthesize those very delicate ratios, and it's hard to identify them from the very noisy sequencing data. And this is why all the examples we're going to see is based on a very low resolution composite alphabet. So what we did, what did we do? We actually took a bilingual version of the Bible. We used we compressed it, and we used the binary file, encoded it into a 6-letter alphabet containing only 2 composite letters, M and K. So we have a 6-letter alphabet. We only added 2 letters to the standard 4 letters. This was mainly because of the technical limitations of the synthesis company. They couldn't support more than 2 extra letters at this point. And we see that even just those 2 letters gives us a 25% increase in the logical density. So we took the same data that was comparable data with comparable parameters to the data that was done in the state-of-the-art work before ours, and we compared the logical density that we achieved, and we got a 25% increase, meaning we reduced the costs of this system by 25%. But we said that we're going to pay with physical density. So what we see here is a dilution experiment. So what we took the DNA that we received from the DNA synthesis vendor, and then we took 10 nanograms of it and went over a series of dilution experiments in which each time we diluted it 10 times, and we sequenced the output to see if we can still observe all the sequences in them, observe perfect copies of them, or maybe infer perfect copies of them, including the composite letters. So we managed to infer the correct ratio for all of the positions, and decode the original information, the Bible in this case. So we can see that the physical density that we achieved was somewhere around 6 petabytes per gram, which is still huge, still orders of magnitude higher than what known today to be in magnetic tapes. But it's somewhere in the low range of what is common today in different DNA storage work. So the highest that people got so far was somewhere around 200, and most people do somewhere between 2 to 20 or 30 petabytes per gram in the physical density. Okay, so maybe we'll talk a little bit about the errors that Amit asked about. So we have to keep in mind that overall we synthesized and sequenced almost 1.5 million oligos. Each one of them was 150 bases long, and roughly one of each 6 letters is an M. So we have hundreds of millions of positions in the design that should have been M, and we can observe all the ratios of A's and C's that we saw in these positions. What you see here that the mean or the average ratio that we got was not a 1-to-1 ratio. We are not on the 50%, but rather tilted a little bit towards C. This is just a bias in the synthesis mechanism, apparently. And you can also see that most of them are pretty close to it, but there is a significant amount of positions in which we read a ratio which is far away from a 1-to-1 ratio. It's still not a very big idea, very big problem when we only have to choose between A, C, and M. But if we wanted to choose between 60-40 and 1-to-1, this would become a very problematic problem. We can see that 6% of the sequence of the positions were actually over the line of being misread as 60-40 and not 1-to-1 in this case. So this is a real concern in terms of going into larger resolutions. Another thing that we have to keep in mind is that sequencing depth matters. So what we did in this case, we subsampled the sequencing reads that we had, and we saw that whenever we have 30 or more reads per sequence, on average, this is enough to recover most of the sequences, and also enough in order to decode the complete information, or the entire message in this case. But what would happen if we wanted to move into higher resolutions? So this is a work based on simulations. We simulated the use of composite alphabets from standard DNA to resolution 10, which is what I described earlier. And we saw that as we go to higher resolutions, the synthesis cost in blue decreases, while the sequencing cost starts to increase. And there is some kind of a sweet spot somewhere around the resolution 5. This is an alphabet size of 56 letters, which represent an overall reduction in cost of somewhere around 50%. Of course, this is just based on simulations. It's based on simulation with experimental parameters estimations, but this is simulated data. And with this, I will summarize the DNA storage part of the talk. So we saw that DNA is an attractive storage media. We introduced the notion of composite DNAs, and how it extends the alphabets and reduces the logical density, while keeping the physical density in a competitive range to the others. And we saw how we can potentially reduce the overall cost of the system up to 50%. I think in this point, let's take 20 seconds in which I will have a sip of water, and you will have a chance to ask questions if you want, before we switch gears to another completely different topic.
Leon?
Hi.
Hi, yes.
It's yours. Yeah, I have a quick question. So you mentioned it like briefly about the sequencing depth, but like the error is not negligible in 30 copies. And if you encode only 200 bases, so you can be ignored, but basically this will lead to an error rate of about, theoretically, 10 to the power minus 6. Like we know it like from like a sequencing depth that is needed for DNA. So what you'll be, so I'm not sure how much it will be efficient when you try to sequence like large data sets, and you need to have like higher depth to get less errors. How can you try to estimate the efficiency of the new letters versus the need to higher depth?
Yeah,
okay. About that.
Yeah, so I mean, maybe you touched a few interesting maybe topics in your question, so maybe let's discuss each one of them separately. Yeah, the need for high sequencing depth is definitely a problem, or a challenge in this case, or maybe a limitation. And we are thinking of ways to maybe deal with it, and maybe use more complicated encoding that will reduce this need. But there are other ways in order to overcome those errors. Specifically, I didn't mention any of them, but we used error correction codes on the encoding data. So even if we do have up to even 10% errors in the read of each one of the sequences, like inference errors in each one of the sequences, we can still recover the original sequence and get away with it. And we do not pay too much in terms of redundancy that we added to the system in order to handle those errors. So maybe the answer to the question is that the solution to the problem is real. We have a problem with sequencing depth, and the solution would come from two different approaches. One would be to think of more complex or more sophisticated encoding, and the other would be add error correction codes on top of the original encoding. So we cannot change the way biology acts. We can only adapt our system to the technology that we're given and the error rate that we're given. We cannot change the way you sample sequences from the tube, and we cannot change the error rate you get from sequencing. We just have to come up with ways to handle it. This is the best answer I can give to this kind of questions, I guess.
Thank you.
Okay, so with that, let's move on to the next part of the talk, which will be much shorter, I promise. What we see here is what we're going to discuss here now is the work that was also done in collaboration with the lab of Roy Amit and with Lior Levy, who used to be a master student in Roy's lab. Hi Lior, I guess you're here. I saw you at the beginning. And we'll start off with a little bit of motivation and background about this type of experiments, because I think many of you in the audience, especially you from the biotechnology department, will find it very useful for their own needs, your own needs, and then we'll move to describe the actual results that we got with Lior. So how can we use oligo libraries in order to investigate gene regulations? So let's first describe a few questions of gene regulation that we can use or we can tackle. In this case, we're going to look at the regulative elements that come upstream to a gene or upstream to the transcription start site of the gene, and ask what is the effects of each one of the transcription factor that binds the specificity of the transcription factor, how many binding sites of the transcription do we need to include in this case, what happens if we change the location or the orientation or the combination of different factors that can bind them, and then ask what happens to the expression level of this specific gene. If you wanted to design this kind of experiment in your lab, you will probably go back to the IDT page, order a few different variants that includes different combinations of the question in hand, measure each one of them separately, the expression level of each one of them separately, and then try to come up with a model that explains the results. But this is very hard to scale, and the way to scale it up is actually using oligo libraries. So what we see here is that we can combine different questions in the design of one big oligo library, then synthesize this entire library and introduce it into our model organism of choice, then come up with a way to measure the performance or the output of each one of the sequences, of each one of the variants that are included in our library. We have to keep in mind that whatever measurement we're going to use is going to have to include both the output, expression level in this case, and the identity of the sequence that generated it, the variant or the sequence variant that was present in this specific cell. There are two ways described here to do it. The first one would be to take all the cells that we have containing a very different pool of sequences, sort them according to our expression level, for example, and then each one of those into different bins, and then each one of those bins can be sequenced, and the sequences in it can be identified, so we can end up with, for each one of the sequences, a profile that says in which one of the bins, or the abundancy of it in each one of the bins. A different approach would be to use RNA-seq as our output, and in this case, we're going to get the identity of the sequence together with the expression level, which is the amount of RNA in one readout, and just normalize it by the number of copies of DNA we see of this variant in our library. And now maybe move on to the actual project of Lior in mind. We start up with a very interesting observation that came up from a different experiment that Lior did in his master's. So what we see here is a construct in which we have an M-cherry reporter gene and a combination of two regions, regulatory regions that can come upstream to it. One would be the Glen-K promoter, which is a sigma 54 dependent promoter in E. coli, and the other would be the PLAC-ARA promoter, which is a sigma 70 dependent promoter. What we see here is that whenever we have the Glen-K promoter upstream to the M-cherry gene, we get very low or zero expression levels. And this doesn't depend on the fact that if we do or do not have the sigma 54 factor available to bind this sequence. It means that just the presence of this sequence of the Glen-K promoter upstream of the genes blocks or silenced the expression that comes from the upstream PLAC-ARA promoter in this case. And our goal was to try and understand why or what happens. What is the sequence determinants within this Glen-K promoter that actually caused this silencing effect. So what we did, we generated a library of 12,000 different sequence variants based on sigma 54 binding regions. Some of them were just variants of different mutations of this Glen-K promoter, but others were just different sequences to ask different questions. We synthesized this library, introduced it into E. coli, and then sorted our cells according to their expression level and generated an expression profile for each one of the variants, just like I just described. Now, if we take all our variants and sort them according to this, the expression level that we see, what we get is that the variant with the lowest expression level or the highest silencing effect contained many copies or many observations of this specific CT sequence motif that was concentrated also in specific positions in the sequence. But the more we had those CT-rich motifs, the stronger the silencing effect was. And the question was, what is the role of this CT motif in the sequence that actually causes the silencing? So we used a subset of the sequences that we had that are the Glen-K promoter with different mutations to it. As you can see here, every red dot is a mutation in the original sequence. And we can see that whenever we mutate those CT-rich motifs, and especially whenever we mutate the one at the end, we get a rescue of the silencing, so we get high expression level of M-cherry, which is a rescue of the silencing effect. Now, the next step was to validate this result. So what we did, we wanted to show that this is necessary and sufficient in order to get the insulation effect or the silencing effect. So we took the Glen-K promoter and we replaced this CT-rich motif at the end, and we actually got a rescue effect of the expression. So we got high expression of this. And then on the other side, to show that this is sufficient, we took a different promoter sequence, which does not include any CT-rich motif. And we saw that whenever we introduce the downstream CT-rich motif, we get a silencing effect. So this is sufficient in order to achieve silencing in this case. And this kind of work allowed us to use an oligo-library approach in order to pinpoint a specific regulatory mechanism within the Glen-K promoter. And this brings me to the summary of my talk, basically. So what we saw is we introduced the DNA synthesis oligo-libraries. We showed how we can improve DNA storage or improve the logical density of DNA storage using composite DNA. We showed how we can use oligo-libraries to perform large-scale systematic assays to maybe investigate specific regulatory mechanisms. And we also showed how, and we maybe gave you a hint of what else you can do with oligo-libraries in different fields. I'm very happy to discuss this with whoever is interested. We have very exciting new projects on the pipeline of what we can do with oligo-libraries. As you can imagine, probably with CRISPR involved in it, and various others. I will skip this slide in which I describe the different maybe fields that we do in Zohar's lab, because we're running out of time. And with that, I will thank you. I will thank Zohar and the group, Roy and his lab, Twist Biosciences for a very successful collaboration on the DNA storage work, and my funding. And I will be happy to take questions now, I think, for a minute or two.
Leon,
Yes.
this is Chazi.
Hi.
Very nice work. In regard to the city repeat, what is the mechanism behind it? I mean, does it form secondary structure, or could you try and fish out the factors that bind to it?
Yes. So we did some analysis on the mechanism here. What we came up is that it is actually a secondary structure of the RNA that is being formed after transcription. We believe that this CT-rich motif is actually acting as an anti-Shine-Delgado sequence. So it binds the Shine-Delgado sequence and blocks translation at the end. And not only it blocks the translation, we also believe that it causes more rapid degradation of the mRNA molecule. I'm not sure that we have any validation, any experimental validations to the actual molecular mechanisms. This would be interesting to perform.
But you can find it in a natural sequence, or this came out only from your random libraries?
No, and it's very nice. It's just like you were our reviewer number 3. So after the request from the reviewer, we also performed a bioinformatic analysis of different bacterial genomes, looking for these CT-rich motifs that maybe can hint on the maybe evolutionary importance of this mechanism, of insulation mechanism. And we did see more CT-rich motifs upstream to specific families of genes. And we believe that this mechanism might have sort of let's call it a protective mechanism for protecting the downstream gene from being maybe mistakenly transcribed from an upstream promoter. So whenever you have an upstream promoter that transcribed the gene together with the sigma 54 binding site that comes upstream to it, this construct would not get translated, because this insulation effect would
kind of terminator.
Yeah, this act as sort of a terminator. Yeah, exactly.
I have more questions. So one is how much it costs today to write and read the Bible, and the second question is whether the length of the DNA oligonucleotides that you worked, is it the optimal one? Did you do analysis that, because I guess the longer the string is, you have more errors and in reading and writing, but on the other hand, you need less reads.
Yeah. Okay. So in terms of length, we pretty much went to the longest we could get from the company, since and they didn't want to synthesize sequences that are too long, because they were afraid of the error rate that would become too high, and they said, okay, but we cannot charge you for a product that we consider not good enough. So we couldn't actually analyze the optimal length. At the moment, I think we're still at the point where whenever we can get a longer sequence, we'll go for it. And in terms of costs, so again, since this work was a collaboration with the synthesis company, all costs were very weirdly calculated. So I can't really give an exact number for this project, but just to give us maybe an estimate, ordering this many sequences, in this case 1 million sequences, would be a matter of a few dozens of thousands of dollars. One large oligo library would be somewhere around 20 to 30, maybe I think somewhere around 20 to 30 $1,000 to synthesize. So it's not yet, but we have to remember that we can store much more than just one copy of the Bible in this one library. So if I try to estimate one copy of the Bible, how much, or just let's say just 1 megabyte, 1 megabyte would be somewhere around, let's say, $1,000 to write once. And we did not mention editing the information if we wanted, which is not possible in this case, or writing it again if we wanted. All those are problems that other people try to deal with, and this work didn't yet.
Thanks. Any more questions? Okay, so let's thank Leon again. Thanks. Thank you very much. And next week we have Uri's guest.